Hello dear students, a very warm welcome to another live session for AIATS Plus Phase 2 where we discuss the questions and the concept in and around it in which most of you tend to make mistake. I am Snehal, your zoology faculty at Aakash. So since the morning we have been brainstorming and trying to figure out the concepts where there has been lacuna, and I know that you must be tired by now. But when it comes to the NEET preparation, there are yet some important topics that needs to be discussed, and that is why I am here. So we'll continue this session with the topics that pertains to the chapter of class 11th and 12th, and they are biomolecules, excretory product and elimination, locomotion and movement, then we have human health and disease, followed by evolution. So as I have always said that amongst all the given topics, it is biomolecules that holds the most significant importance, because in NEET exam it has been noticed that every year maximum number of questions has been asked from this chapter. Alright, so without wasting much time, let's begin with our discussion. So the first question that we have today in our bucket is question number 136. And it states, a population will not exist in Hardy-Weinberg equilibrium if all of the following are present except. So before reading the options, let us first talk about the beauty of evolution and that particular chapter. So basically how you are trying to basically how you and me, or the tiny bug around you, or the giant elephant that you see has come into existence is because of this stochastic process known as evolution. So basically, evolution, the word itself means that to evolve, to form something. Alright? So basically, how we came into existence? It was because of evolution, and that is what the two renowned scientists, Hardy and Weinberg, tried to explain by giving a mathematical proof. So basically, the Hardy-Weinberg equilibrium is a mathematical proof of the fact that evolution is continuously occurring to bring life into force. Alright? So, what these two scientists stated, that if evolution is not occurring in a given population, then the gene pool of that particular population will maintain an equilibrium. I can see students have joined, and a very good afternoon to everyone. I hope you enjoy the class. So let's continue. So basically they said that the gene pool of a particular population which is experiencing no evolution will remain constant. So gene pool remains constant, or it will remain in equilibrium. What is gene pool? Gene pool is basically the sum total of all the allele frequencies in a population. This is the sum total of all the allelic frequencies in a population. Alright. So where is the mathematics? So the mathematics comes here into picture is that they stated that P plus Q will be equal to 1, which is the constant number, where P represents the allelic frequency of the dominant trait, whereas Q will represent the allelic frequency of the recessive trait. So basically, the sum of P plus Q in a population should be equal to 1. This is what Hardy-Weinberg were trying to explain, that in ideal conditions, it is never possible that the sum of these allelic frequencies will be equal to 1, which eventually means that evolution is a continuous process which is occurring, and therefore they further implied that we can assume that these conditions will exist only if there are certain factors which are not operating in a particular population, and those factors are said to be the disturbing factors that will affect the equilibrium of a population. So we'll come to those factors, but before that, let us expand this equation by applying binomial theorem, and the equation that we'll get is P plus Q square plus 2PQ is equal to 1. So the difference that you need to remember even while solving the numericals which we get from this topic is that P represents the allelic frequency of the trait, whereas P square will represent the frequency of the genotype. To make it simpler, let us assume that we have a trait for tall tree. So here allelic frequency will mean that we are only talking about T in a population, whereas if we talk about P square, it means that we are talking about T and T. All right? I hope it is clear so far. Please give me a thumbs up in the comment section if you feel that it is making sense to you. All right? So P square here is the frequency of the genotype of the dominant allele, and Q square is the frequency of the recessive genotype of the recessive trait, whereas 2PQ here represents the frequency of the heterozygotic condition, which means T and T. And in the comment section, I can see that someone has commented homozygous dominant frequency. And dear student, you are absolutely right. We call this as homozygous dominant frequency because we have two capital letters, or to say it is a homozygous condition, similar condition. Whereas here, as I stated, it is a heterozygous condition. Now moving to our main discussion, that what are the factors that Hardy-Weinberg talked about, that if a population has to stay in equilibrium, these factors should not be present. So those factors were, the first and foremost, gene flow, then we have natural selection, also we have mutations, then we have genetic recombination further we have another factor which is selective mating. It is random mating which should be present. Random mating will favor this genetic equilibrium. So basically selective mating should be absent. So basically if all these factors are present in a population, then the population will not follow the Hardy-Weinberg equilibrium. Alright, so let us discuss these factors quickly. So the very first factor that we have is gene flow. So basically gene flow means flow of or the exchange of gene frequency, and it happens when there is migration of that particular gene frequency, which means for instance that we have a country, for instance let us take that we have USA, and if some individuals are entering the country, it means that from the original area there will be some loss of the gene frequency, and here in USA there will be some gain in terms of gene frequency. So this is going to disturb the gene pool of both the area, because from the original place there will be loss of genes, whereas in USA there will be gain of genes. And a similar thing will happen when people will move out from that particular area. For instance, if someone moves out of USA, then now USA will experience a loss in gene frequency, whereas the person moving to the other place will experience a gain in that gene frequency. So we can see that if there is this gene flow, then the population is not going to follow the Hardy-Weinberg equilibrium, because there is constantly change in the gene pool of that particular population. All right. So now we move on to our next factor, which is mutation. So basically mutation is said that it provides the raw material for evolution. So basically what happens is that if an organism is undergoing mutation, then when natural selection will operate, then the organism which will be most favored in a particular given condition, the mutation occurring in that organism will favor the survival of that particular organism. So basically, let's suppose that we have a mutation which is favoring which is causing an organism to survive in harsh condition like cold weather. And for some instance, this mutation occurred very randomly, like before the condition of cold weather arrived, these mutations were hidden in that organisms, which means I'm trying to say that these mutations were yet not expressed. But if there is a condition like cold weather, then in a colony of organisms, which have these mutations will survive, and the organisms which do not show these mutations will die. And this is how natural selection operates. So nature will favor that organisms having these mutations should evolve because they can survive better in a given environment, and that is why we call it that mutation is providing raw material for evolution, because that particular trait in the organism was hidden in the form of mutation until and unless they were not exposed to such harsh condition. And for this we have a very famous experiment done by Lederberg and Lederberg, where they expressed that, where they showed through experimentation, they showed that in a population of bacterial colonies, they were trying to grow a population of bacterial colony. So they showed normal growth, but after some time they added some antibiotic resistance. So they added some antibiotic to it. All right. So when they added some antibiotic, it was observed that out of the given colonies, there were only few colonies which survived further, whereas the rest of them died. So this explains the fact that some sort of mutation was already present in the population here, but it was not being expressed until and unless an external factor started operating. All right, and then this external factor led to the expression of these mutations which were favored by natural selection, and further natural selection favored the survival of these particular colonies. All right. So basically we are trying to say that natural selection will also affect the genetic equilibrium in a population, because natural selection is operating to eliminate a particular gene, which is not suitable to survive in an environment. So natural selection is also disturbing the genetic equilibrium. All right. So we have discussed three factors so far: gene flow, migration, natural selection, and now we have the other important factor, which is selective mating. So basically, in a population that is exhibiting selective mating, that population will not be in Hardy-Weinberg equilibrium, because if selective mating is being performed, it means that only a particular gene is being selected and is being favored. For instance, if selective mating is being performed only between two trees that show tall characteristics, it means that in that particular population we will have only tall trees, and this is how the short trees are getting eliminated. And thus this shows that for Hardy-Weinberg to operate, random mating should be exhibited by the population. Now coming to another important factor, which is the genetic drift. Please let me know in the comment section, what can you recall when you hear genetic drift? There are two very important phenomena we talk about when we talk about genetic drift. All right. I can see someone in the comment section has mentioned that Joshua and Esther Lederberg. All right, so these are the names of the scientists, and thank you for your input. Okay, so further we discuss genetic drift. So genetic drift is basically a condition where there is a sudden loss of gene. For instance, we can study genetic drift through two effects, which is known as the founder effect and the bottleneck effect. So basically what happens in founder effect is that, let us imagine that we have an island where we have two populations, AA and BB. Due to some competition here, it was difficult for the survival of BB. So what they did, they migrated to another new area and they started their population in that new area, and this is how this migrated population will be known as the founders, and this effect is known as the founders effect. And a classical example of this is the Darwin's finches. We all know that Darwin's finches started facing competition in South America, and then they traveled to Galapagos Island, where a seed-eating Darwin finch migrated to Galapagos Island, and here they started their own population, and then they diverged into different species, and they all had a particular type of beak shape that helped them eat different substances. For insect, they started eating fruits, some ate cactus tree. All right, so basically what happened here is that the population of Darwin's finches moved to new place. So here there was loss of genetic diversity, but here it was gain of genetic diversity. So basically we are trying to imply that whenever there is this disturbance of gene, it will lead to loss of equilibrium. All right, then we have the bottleneck effect. So basically bottleneck effect tries to explain that, for an instance, we have a population of beetles in a grass where we have two types of population. One is the green colored beetles and the other one is the blue colored beetle. So what happened is, it was observed that suddenly the green colored beetles died. And how they died? It was found that these green colored beetles died because there were elephants who rampaged on them. And why that happened? It was because they could not see, because of the green grasses around, that there are green beetles present. And this led to the remaining beetles to be blue colored only. So basically we are calling it a bottleneck effect, because if you see a bottle which was full of different types of beetles, for instance, is now left only with these blue colored beetles. So basically there is a loss of genetic diversity. All right. So this is how these different phenomena are going to disturb the Hardy-Weinberg equilibrium. So let us quickly go back to our question now. So our question states that presence of which of the following factors will not let the population exist in Hardy-Weinberg equilibrium. So we discussed that gene migration, mutation and genetic drift, all three of them can lead to loss or gain of genetic equilibrium, and therefore our right answer will be random mating, because if selective mating is performed, then a particular trait only will be favored for in that particular population. Alright, so now we move on to our next question. And again we have a similar question where it is asking that according to Hardy-Weinberg principle, the frequency of AA individuals in a population is denoted by. So I would like the answer from you, since I have already discussed this very thoroughly, that what should be the frequency of AA population. Please let me know in the comment section. So the answer should be option third, P square, which represents the frequency of homozygous dominant allele. I can see students answering, and you are absolutely right. So now we move on to our next question, which is a match type question, and it states match column 1 and column 2 and choose the correct option, where under column 1A I can see that there are names of different bands, and under column B I can see that they are talking about the constituents of that particular band. Alright. So now quickly move to the discussion of what these bands are. So basically we all know that our muscles are made up of various muscle bundles, and these muscle bundles are also known as fascicles, as our NCERT states. Alright. Further, our muscle bundles, if we do a microscopic analysis, they are said to be made of muscle fiber, which is also known as the muscle cell. Further, if we again zoom in more deeper into the structure, then muscle fibers are said to be made up of myofibrils, and myofibrils are further made up of myofilaments. And myofilaments are said to be two types of myofilaments, which are known as the thick and the thin myofilaments. Alright. So basically from muscle we came to myofilaments, and we have thick and thin myofilaments, and let me know now in the comment section what are the specific names of this thick and thin myofilaments. So we further move to our discussion, and here you can see a structure, which is a diagram which is taken from the NCERT, and this is a diagram of a myofilament, and we can see that we have the thick and thin myofilament that I talked about, and that thick and thin myofilament here is actin, which is the thin myofilament, and then we have myosin, which is the thick myofilament. Alright. So if we see the structure of this myofilament, there are few important inferences that we need to draw, and it is that first of all we can see that we have one A band. Alright. So A band is basically the length. A band represents the length of this myosin. So we can also say that A band represents the length of myosin, and it is made up of both actin and myosin. So its structural component will constitute both actin and myosin. Further, since it is containing both actin and myosin, it is known as the anisotropic band. Aniso means uneven. So here the uneven factor is the actin and myosin, all right? So we have seen our A band. Now let's move to the I band that we see in our structure. So I band is basically the distance between the two myosins. Here we can see that I band is the distance between the two myosine. And from the image, we can also see that I band contains only the actin filaments. So our I band will contain only the actin filament, and that is why they are called as the isotropic band, which means that they are similar in the sense that they constitute only one element, which is the actin. All right. So now we move on to our next labeling, which is the Z line. So Z line is basically one elastic, I would say, fiber which is bisecting the I band. You can see that it is passing through the I band. So Z line is passing through the I band. So we can say that it is bisecting the I band. So from this few words that you see, we get many questions when it comes to NEET exam, where you can be asked things like that I band constitutes actin or myosin or both. So we all now know the answer, that it is made up of actin only. And then further we can be asked that which line bisects the I band. So answer here will be the Z line. Okay. I can see Srinath is asking that A band contains both thin and thick filaments, and it is absolutely true that A band will contain both thick and thin filaments, and this is how you can be asked question in the NEET exam as well, that whether thin and thick filament are present in A band or not. Now further, if we analyze the diagram, we can see that these Z lines are being held in place with the help of the actin filament. This is again a point where question is being asked, that Z line is held in position with the help of actin or myosin. So our answer will be actin. All right. Further, as we discussed in the previous slide, that our A band represents the length of myosin, and this is why we say that when sliding occurs or contraction occurs, then the length of myosin will not change, because when contraction occurs, this band will slide, the thin band will slide over the thick filament. All right. So basically my actin will shift and the myosin filament will remain at its place, and then there will be no change in its shape. Whereas if this actin is moving forward, then obviously the length of this Z line will change, because these two fragments will come closer. Also our length of I band will reduce, because this I band will be pushed inward. All right. So these are some of the key concepts from where question is being asked in the NEET exam. We can see another labeling which says the H zone. So basically when you see the arrangement of actin and myosin, we can see that there is a zone where there is no overlap. Overlap in the sense there is no actin present here. So basically this unoverlapped zone is known as the Henson's node. and the length of Henson's node decreases during muscle contraction. Also, our sarcomere will also decrease in size when contraction occurs. Because the Z lines will move closer to each other. All right? One important concept here again is sarcomere will be made up of which two proteins, and the answer will be that the sarcomere is made up of, we can see a sarcomere is made up of half of I band. Here we can see we have the half of I band from this side. And we have the complete A band which is present in the, which is constituting the sarcomere. So basically a sarcomere is made up of two half I bands of either side and one A band. And this is a very favorite question which is often asked in the NEET exam. So the sarcomere is composed of half of I band plus the A band, and then we have half of I band from the other side. So now I'll quickly go back to our question. So we can see that I band will match, since I band contains only the actin filament, so our option 2 states has only actin filament. So A will match with 2. A band contains both actin and myosin filaments. Therefore, B will match with 3. H band has only myosin filaments, as we discussed that there is no overlap. So H will match, C will match with 1. And finally, we have the Z line, and it is a membrane to which actin filaments are attached, and we saw in the structure that actin filaments were attached to Z line to hold it in place. So D will match with 4. And the right combination of matches that we see in our options is our option number 2. So our right answer will be option number 2, and we'll straight away eliminate the other options. Let me know in the comment section that whether actin is known as the regulatory proteins or they are called the contractile proteins. So now we move on to our next question, which is question 154. And it states, from given list of biomolecules, how many of them are present in retentate during analysis of living tissue? All right. So among the options given, we need to find out that which molecule will be present in the retentate. And I can see someone has answered in the comment section that actin is a contractile protein, and you're absolutely right. Thank you for your answer. So basically, we all know, like right now I talked about evolution. So we all know that how life came into existence. Life came into existence by aggregation of small non-living molecules which gave rise to us, a huge animal, right? So basically, our body is composed of many non-living molecules or elements, and this is the beauty of biomolecules, which means that, which states that non-living molecules come together to form a living molecule. So to perform chemical analysis of what we are made up of, a general procedure is followed where a tissue is taken. Obviously, we'll take a living tissue. We'll take a tissue sample from a living organism, and then it is grinded with the help of mortar and pestle to form in the presence of trichloroacetic acid to form a thick slurry. Just imagine that you are crushing a substance, so obviously a thick slurry will be formed. Now this composition that is used here, which is the trichloroacetic acid, is an important part because questions are often asked that which acid is used to grind living tissue, alright. So the thick slurry now obtained is made to strain through a cheesecloth, and as we strain it, we will get two fractions. One will be the acid soluble fraction and the other one will be the acid insoluble fraction. Alright. So it is just like that. Imagine that you're in the kitchen and you're trying to make tea, and when you strain the tea, the liquid part will go inside the cup, and that is the acid soluble part here. Whereas just imagine you have added cardamom and ginger to your tea, so those two bigger fragments will stay over the sieve, alright. So that part will represent, the part which will stay over the sieve represents my acid insoluble fraction, and they are also called as the retentate and the filtrate. The retentate and the filtrate. All these terms are mentioned in the NCERT, and questions are often asked around these terminologies. So it is important that we are clear with these small definitions. This is called as the filtrate. Also, we also name it as the macromolecular fraction, whereas the other one represents the micromolecular fraction. Alright. So now let us quickly discuss the acid soluble and the insoluble fraction. Alright. So as I mentioned that here we are getting the macromolecular fraction. So it is obviously implied that the substances present in this filtrate will be made up of will have molecular weight which ranges around 10,000. Alright, whereas when we talk about the acid soluble fraction, here the molecular weight normally ranges around 1800 to 800 Dalton, I'm sorry. Now, in this also we have an exception. So basically the substances which are found in the acid soluble insoluble fraction are the polysaccharides, then we have proteins, we have nucleic acids, and we also obtain lipid in this molecular fraction. Now, why lipid is important and should be studied separately is because of the fact that lipid is not a polymer. It is not a polymer, and its molecular weight is usually less than 800 Dalton. But still, why is it present in the acid insoluble fraction? This is what this question should intrigue you, that why, since we saw that the molecules which were found in the acid soluble fraction had this particular weight range. So basically हमें acid soluble वाले fraction में lipid मिलना चाहिए था, but that doesn't happen. And why is that? Because when we break down a cell membrane, we all know that our cell membranes are composed of lipids. We call it a lipid bilayer, right? So when we were making that thick slurry and grinding, so these plasma membrane will break down into small fragments or small vesicles which are water insoluble vesicles. And why did that happen? It happened because lipid has that hydrophobic nature, which means it will not solubilize in water. So basically because of that lipid, even though it is not a polymer, even though it has molecular weight less than 800 Dalton, it is found in the acid insoluble fraction. Alright, I hope I am making sense. So now we move on to our acid soluble fraction. So basically our acid soluble fraction will comprise of substances like amino acids, the monosaccharides that we have, and the nucleotides and the nucleosides. Why? Because they have less molecular weight, so basically they get filtered out. So now let us go back to our question, and our question was asking that among the given molecules, which one will be present in the retentate. So basically we are talking about the acid insoluble fraction. So when we read the molecules, we will try to figure out the nature of that particular molecule. So we all know that keratin is a protein and it is a polymer, and therefore it will be found in the retentate. Therefore we will include keratin in our answer. Coming to nuclease, we say that nuclease is an enzyme and obviously it is also a polymer. Therefore we will include nuclease in our answer. Alright. Then we have glycine. So glycine is basically said to be the most simplest amino acid, and often question is also asked on this, that which is the simplest amino acid, and that is what our answer will be. It is glycine which is the simplest amino acid. Alright. So obviously since it has low molecular weight, it will not be found in the retentate, therefore will not include glycine in our answer. Then comes palmitic acid, and we all know that palmitic acid is a lipid which is a 16 carbon fatty acid, and as we talked that lipid will be present in the acid insoluble fraction, therefore will include palmitic acid. I can see that Srinath is asking whether retentate contains macromolecules having molecular weight more than 10,000 Daltons. So the answer is right. Yes, the retentate will contain molecules that have molecular weight more than 1000 Daltons. Alright. Now moving to glucose. Glucose, again we discussed in a previous class very clearly that glucose is a monosaccharide. Alright. So glucose is a monosaccharide, and therefore glucose will not be found in the acid insoluble fraction. Then we have cytidine. So cytidine is again a nucleoside, and nucleoside will not be found in the retentate, therefore will not include cytidine in our answer. So we can see we have just 3 molecules that will be found in the retentate, and among the options given we have 3 in our option third, therefore answer will be option third. Okay, so we move on to our next question now, and it again pertains to the chapter of biomolecules, and it states all the given statements are correct except. So basically among the given options we need to look for the one which is incorrect. So our first statement states in a polysaccharide chain the right end is the reducing end and the left end is the non-reducing end. Now, first of all, to understand what is reducing and non-reducing end, let us talk about the concept of reducing sugars and non-reducing sugars. So basically, a reducing sugar is the sugar which contains a free aldehyde or keto group where the carbonyl part of the aldehyde and the keto group, the carbonyl part of the aldehyde and the keto group can participate in redox reaction, meaning to say this carbonyl will act as a reducing agent. It means that carbonyl will be able to lose an electron or gain an electron, all right. So basically, for a sugar to be called as reducing sugar, ये carbonyl group or free aldehyde or ketone group should be present in the structure of that sugar. Coming to non-reducing sugars, so basically it's just the opposite, that free aldehyde or ketone group is not present. So basically, these molecules or these sugars do not have a carbonyl group or a group which can act as the reducing agent, and therefore these sugars act as the non-reducing sugars, all right. And how do we get to know that whether a particular carbohydrate is reducing or non-reducing? It is based on the fact that we perform the Felling's test. So basically what happens in Felling's test is that copper is reduced into copper ion, where the blue color of copper will change into a red color, and this indicates that this particular sugar, if a sugar is giving a positive Felling's test, it means that that sugar is reducing in nature, all right. And when we talk about polysaccharides, let us take the example of the structure of glycogen. We call it that the left end of a chain of glycogen is the non-reducing end. It is the non-reducing end, whereas the right end is called the reducing end, and it is called so because here we can see that it is the reducing end, because here we see that there is no free ketone or aldehydic group to participate in forming a bond, whereas the right end participates in forming a bond, and therefore this helps us conclude that our first statement is correct, and therefore option 1 cannot be the right answer. Now moving on to our option 2, which states starch can hold I2 unlike cellulose. So when we talk about the structure of starch, we say that it is made up of amylose and amylopectin, where amylose has a helical structure, whereas if you see the structure of cellulose, it is a simple structure where there are no helices, whereas in the structure of starch, helices are present. And why it is present? It is present because of the amylose, and thus what happens is that I2 molecules get trapped in these helices and give a positive test, which means a blue-black colored complex will be formed when I2 and starch is made to react together. All right. So this also brings us to the fact that our second statement is correct and therefore it cannot be the right answer. Now moving to our third option, which states chitin present in exoskeleton of arthropod is a homopolymer. So it is absolutely true, as we all know, that arthropods have a chitinous exoskeleton which is made up of an acetyl glucose amine. So basically this chitin is a polymer, but it is a polymer of a single sugar, which is the N-acetyl glucose amine, and therefore we say that chitin is a homopolymer, whereas if a polysaccharide is made up of more than 2 sugars, they will be called as heteropolymer. For instance, we know that peptidoglycan present in the bacterial cell wall is made up of N-acetyl glucosamine and N-acetyl murmuric acid, and therefore our option third is also correct. So finally we move to our last statement, which states polysaccharides are found in acid soluble fraction. So we already discussed it very extensively in our previous question, that polysaccharides will be found in the acid insoluble fraction and not acid soluble fraction, and therefore the answer that we are looking for is option 4. So this marks the end of our discussion and I hope it was fruitful. Hope to see you